Good afternoon. I'm Kevin Grady, the Senior Product Line Business Manager at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series. Today's webinar, entitled iPSC-derived primary cells, expand your cell-based assays with an unlimited and biologically relevant resource, is presented by Dr. Brian Shapiro. Brian Shapiro is a scientific content specialist for cell biology at ATCC. In today's webinar, Dr. Shapiro discusses how differentiated induced pluripotent stem cell-derived cells fulfill the need for a consistent source of cells and physiological relevance. He will then provide data demonstrating how differentiated iPSC-derived cells can be incorporated into immunoassays or further differentiated into other cell types, such as osteocytes, chondrocytes, and adipocytes. If you have any questions for our speaker, please use the chat function available through the webinar program. Questions will be answered as time allows at the end of the presentation, and any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website at www.atcc.org. So with that, I would like to welcome Dr. Shapiro.
Thank you, Kevin. Before I jump into my talk, I'd like to say a quick word about ATCC. Founded in 1925, ATCC is a nonprofit organization with headquarters in Manassas, Virginia, and an R&D and services center in Gaithersburg, Maryland. We are the world's largest, most diverse biological materials and information resource for cells and microbes. We are also an innovative R&D company featuring advanced in vitro models such as iPSC-derived primary cells, which is what I will be discussing today. So today I'll start by talking about how to select the appropriate cell line for your experiments. I'll quickly run through where differentiated induced pluripotent stem cells, or iPSCs, lie in the continuum of cell models. Then I will give some background about iPSCs. I'll go into ATCC's portfolio of iPSC-derived cells, showing you their differentiation potential and some application data. I'll then end my talk with a wrap-up of the presentation and take your questions. So let's jump in. First, I'm going to discuss the pros and cons of the different cell models and where ATCC's new iPSC-derived cells fall. ATCC has a large variety of classical and advanced cell models. We developed these advanced cell models to enhance our offerings and continue to be a complete solution provider. Most people are familiar with ATCC for our continuous cell lines such as HeLa cells and A549 cells. But as you continue down your research path, you will need more biologically relevant cell types. And ATCC provides these in the form of primary cells, H-tert immortalized primary cells, and now iPSC-derived primary cells. All these cell types feature ATCC's cell line reliability, authentication, and performance. So like I mentioned, I first wanted to talk about selecting the appropriate cell model for your research. As a researcher, one of the first questions you have to ask yourself is, which model is the best for my workflow? This table breaks it down for you. Let's start with continuous cell lines. They have high proliferation capacity, and they are sturdy cells that are easy to work with, but they are tumor-originated cells, which make them less biologically relevant. They might have chromosomal aberrations or mutations or things like that basically make them lose some of their physiological relevance. By contrast, primary cells are isolated directly from tissue, so they have the greatest biological relevance. However, the supply for primary cells is relatively low from a given donor, and there can be significant lot-to-lot variability due to biological variance between the donors. Now next, there's H-tert immortalized primary cells. These cells are genetically modified primary cells. H-TERT cells exhibit normal primary cell physiology, but have the growth characteristics of a continuous cell line. They have near normal genetic stability. They have the ability to generate a large number of cells for large drug screens or toxicology studies, providing limited to no donor variability. And then last, the star of today's presentation, iPSC-derived primary cells. They significantly mimic in vivo tissue type physiology and genetic stability, and we can generate a great number of production lots from an individual clonal source, which highly limits their lot-to-lot variance. Now, just to mention, as a complete solutions provider, ATCC offers mesenchymal stem cells in primary H-TERT immortalized forms. These are isolated from adipose tissue, and also an iPSC-derived version from bone marrow. We offer CD34 positive cells in primary and iPSC-derived, isolated from bone marrow, and we have monocytes in primary form, isolated from peripheral blood, and iPSCs derived from foreskin fibroblasts. The differences here are the primary cells will need to be used immediately and are not amenable to passaging. The H-TERT immortalized cells can be passaged to over 25 PDLs, or population doublings, post-thaw. They're derived from one donor and thus show reduced interexperimental variability, like I mentioned before, and they represent one solution. However, the H-TERT immortalized solution isn't available for each cell type. iPSCs are another solution. As I mentioned, they're also derived from a single donor, but either can be terminally differentiated or used right out of the vial. And, as I mentioned, these will exhibit the greatest interexperimental reproducibility. So I've talked about where iPSCs fall in the research workflow. Now I'll introduce iPSCs and talk about what iPSC-derived cells are and what they can do for your research. iPSCs, which, as I mentioned before, stand for induced pluripotent stem cells, are derived from somatic cells that have been reprogrammed back to an embryonic-like pluripotent state. These iPSCs can be subcultured indefinitely as long as differentiating cells are removed from the culture. Alternatively, iPSCs can be terminally differentiated to any cell type via various culturing methods with lineage-specific media formulations. Any cell type is possible, from ectodermal lineage cells, such as neurons and keratinocytes, to mesodermal lineage cells, such as hematopoietic and endothelial cells, to endodermal lineage cells, such as thyroid cells and the acinar, ductal, and endocrine pancreatic cells. To generate iPSCs, ATCC introduces the reprogramming factors OCT4, SOX2, KLF4, and MYC genes into somatic cells using a Sendai virus integration method. This method is footprint-free, producing a final product that does not contain any viral DNA. The method provides a renewable source of pluripotent stem cells that can then be turned into the cell type of your choice. While iPSCs are useful in and of themselves, many researchers prefer to have their cells in a ready-to-use format. We've also heard many toxicologists and cancer researchers were challenged by the lack of a consistent source of cells with high physiological relevance for their cell-based assays. The source tissue for primary cells comes from a variety of donors, which can introduce interexperimental variability and confound experimental results. Cell lines, by virtue of the fact that many of them are from neoplastic tissue, may not have the physiological relevance needed to make the assay predictive. So ATCC heeded this call and produced cells that answer all of these needs. So let's talk about the star of today's show, iPSC-derived primary cells. Like I mentioned, ATCC wanted to create a versatile cell type that could be produced in great quantities, yet retain physiological function. We looked to in-house produced iPSCs as feedstock for this project, as they were, one, available, and two, we have over a decade of experience culturing these cells. For differentiation, the iPSC-derived cells were differentiated to the desired cell type via incubation in proprietary media formulations. Also, since we used in-house produced iPSCs, we can supply the parental iPSCs, which can be used as a control if needed. The major benefit of using a single clone source to generate each of these iPSC-derived primary cells is that you have the potential for an unlimited, consistent source of cells, which you can use for large-scale research applications, whether it be research or therapeutic purposes, and suffer no donor variability. For the first phase of the portfolio, ATCC decided to focus on 3 cell types: mesenchymal stem cells, CD34 positive progenitor cells, and monocytes. To bring us all up to speed, I thought I'd go through some definitions. So let's start with mesenchymal stem cells, or MSCs. These are multipotent stromal cells that can differentiate into a variety of cell types, including osteoblasts, or bone cells, chondrocytes, or cartilage cells, myocytes, or muscle cells, and adipocytes, which are the fat cells that give rise to marrow adipose tissue. Next, we have CD34 positive cells. These are hematopoietic stem cells that can differentiate into the cellular components of blood. For example, erythrocytes, or red blood cells, leukocytes, or white blood cells, and platelets. Finally, we have monocytes. Monocytes are a type of leukocyte that can differentiate further into macrophages and dendritic cells. Now let's take a deeper dive into ATCC's new iPSC-derived differentiated primary cell portfolio. The first product I'd like to talk about is iPSC-derived mesenchymal stem cells, ATCC part number ACS-7010. These cells are derived from ATCC's bone marrow iPSCs. They show high purity, as 95% or greater of the cells are positive for a number of markers such as CD29, CD44, CD73, CD90, CD105, and CD166. These are checked by flow cytometry. Less than 5% of the cells are negative for CD14, CD19, CD31, CD34, and CD45. In addition, less than 5% of the cells are positive for the iPSC marker TRA160. Now, these cells do exhibit high post-thaw viability at greater than 90%. Since they come from a single clone, they will be available in large quantities. And they also show high osteocyte, adipocyte, and chondrocyte differentiation potential. I should note that we are using serum-free media to cryopreserve these cells. So as I mentioned, we use flow cytometry to monitor the surface markers of the iPSC-derived MSCs for QC testing. These data show that expression. On top of the standard MSC markers of CD73, CD90, and CD105, we test for CD29, shown on the upper right, CD44 on the upper middle, and CD166 on the lower middle to meet International Society for Cellular Therapy guidelines. In order to further characterize that over 95% of the initial starting material differentiate into MSCs and not any other cell type, our release testing includes MSC negative surface markers. And here I'm showing you that. Very low expression of CD14, CD19, CD34, CD45, and TRA160. By the way, this information is also provided in the lot-specific C of A. As I mentioned before, MSCs have the potential to differentiate into mesodermal lineage cells such as bone, cartilage, and fat cells. The next couple of slides show this. Here, our iPSC-derived MSCs were differentiated to osteocytes using ATCC's osteocyte differentiated tool, part number PCS-500-052. We did this for 37 days and stained using alizarin red, which stains bone cells. Undifferentiated MSCs are shown at the left image on day 37, and differentiated stained cells are shown on the right. The red staining indicates the presence of MSCs differentiated into osteocytes. Here, iPSC-derived MSCs were differentiated to chondrocytes in 3D culture using chondrocyte differentiation tool, which is ATCC part number PCS-500-051. For 21 days, we did that. The cells were fixed, sectioned, and stained with Alcyon blue stain, which dyes cartilage. I used the control from the previous slide for comparison so you can see the profound blue stain indicating the cartilage in the extracellular matrix. In this slide, iPSC-derived MSCs were differentiated to adipocytes using an adipogenesis differentiation kit. We did this for 21 days and stained using oil O red stain, which stains fat cells. Undifferentiated MSCs are shown in the image on the left, and differentiated and stained cells are shown in the image on the right. So these data indicate that the iPSC-derived MSCs can be differentiated into 3 different cell types. MSCs have also been reported to immunosuppress activated peripheral blood mononuclear cells. ATCC has previously shown that with bone marrow-derived MSCs. Here, either iPSC-derived MSCs or primary bone marrow-derived MSCs were co-cultured with peripheral blood mononuclear cells, or PBMCs, at different ratios and evaluated for CD45 PBMC using flow cytometry. Red bars represent iPSC-derived MSCs, and the green bars represent bone marrow-derived MSCs. As you can see in the control, where there are no MSCs, most of the lymphocytes are proliferating and are expressing CD45. When the PBMC to MSC ratio is 1:1, most of that proliferation is arrested, and this is seen in both the bone marrow and iPSC-derived MSCs. Even after increasing the PBMC to MSC ratio, you can still see that PBMC proliferation is inhibited. I'd like to point out that lot-to-lot variability was significantly higher in the bone marrow-derived MSCs than the iPSC MSCs, indicated by these error bars. This supports our claim that iPSC-derived MSCs provide more consistent results or better interexperimental consistency. So I've showed you that the iPSC-derived MSCs can differentiate into fat, bone, and cartilage cells, and they can also immunosuppress activated PBMCs. So now I'll discuss the second product in the portfolio. The second product is iPSC-derived CD34 positive progenitor cells, which are derived from ATCC's bone marrow iPSCs. The part number is ACS-7020. These cells show high purity and post-thaw viability. They are suspension cells and are available in large quantities. As CD34 positive cells, they show high erythroid, myeloid, and megakaryocyte differentiation potential. They're also cryopreserved in serum-free media. So here's the flow analysis of the iPSC-derived CD34 positive cells. Positive markers that we test for are CD34 and CD45. For CD45, even though it's a positive marker, it needs to be below 80% to show that the cells are not differentiating too far. The negative marker, TRA160, is a stem cell surface marker. You can see that CD34 is over 90% and TRA160 is less than 1% here, indicating the high purity of the cells. In the next study, I'm about to show you the differentiation potential of iPSC-derived CD34 positive cells. In this slide, I'm showing you the general path of differentiation that a hematopoietic stem cell can take once it's been treated with the right cocktail of hormones and chemicals. When incubated with the appropriate media, iPSC-derived CD34 positive cells differentiate into colony-forming unit that generates myeloid progenitor cells, aka CFU-GEMM. The GEMM, by the way, stands for granulocyte, erythroid, monocyte, and megakaryocyte. These further differentiate into a mixed bag of progenitors. Megakaryocytes, which give rise to platelet progenitor cells and platelets. BFU-E, or burst-forming unit erythroid, which lose their nucleus, produce hemoglobin, and differentiate into CFU-E, or colony-forming unit erythroid, which give rise to mature erythrocytes or red blood cells. CFU-GEMM can also specialize into mast cells. The cells can also be coaxed into becoming myeloblasts, which differentiate into the various granulocytes or other components of the immune system. And so I'm showing you some of that differentiation potential in this slide. Here, the CD34 positive cells were differentiated to erythroid, myeloid, and megakaryocyte lineages using a commercial kit for 16 days. Basically, this kit contains erythropoietin, insulin, and the other factors needed to send the cells down the appropriate lineage. The micrograph on the top left corner is the CD34 positive colony seeded at 15K. Top middle is CFU-GEMM, which generates the myeloid cells. Top right is CFU-GM, which are precursors to monoblasts and myoblasts and are derived from the CFU-GEMM. Bottom left is CFU-GEMM with hemoglobinized erythrocytes, and you can see the red of the hemoglobin. Bottom middle is CFU erythroid, derived from the CFU-GEMM. And finally, bottom right is the burst-forming unit erythroid. So, like the iPSC-derived MSCs, the iPSC-derived CD34 positive cells have high differentiation capacity. All right, I've showed you the first two products in the iPSC-derived portfolio. Here's the final product that we offer, the iPSC-derived monocytes. ATCC product number ACS-7030. These cells are derived from ATCC's iPSC foreskin fibroblasts. Like the other iPSC-derived lines, they have a high purity and post-thaw viability. We see greater than 90% of the cells to be CD14 positive. Like the other iPSC-derived cells, production lots derived from a single clonal source provide for limited lot-to-lot variability. As you can see in the upper right-hand panel, these cells' growth mode is in suspension. In vivo, monocytes can differentiate into dendritic cells and macrophages, shown in the lower panel. Macrophages need almost no introduction. Macrophages are the large white blood cells produced through differentiation of monocytes. They're capable of detecting, engulfing, and destroying pathogens in apoptotic cells. They also can respond to inflammatory stimuli. Dendritic cells, on the other hand, are a type of bone marrow-derived leukocyte. They're the most potent type of antigen-presenting cell of the mammalian immune system. Their main function is to capture antigens and present the processed antigens to the T cells. They act as messengers between the innate and adaptive immune system. They can also be used for anti-cancer vaccine development. As the lower panel alludes, ATCC-derived monocytes show high macrophage and dendritic cell differentiation, along with high cytokine activation and a high phagocytic potential for macrophages. I'll be showing you that over the next couple of slides. So we differentiated the iPSC-derived monocytes into macrophages for 6 days using in-house macrophage differentiation medium. We also used maturation mediums. Here, on the leftmost image, you can see the morphology of the macrophages on day 6. They have attached to the plates and flattened out. In the middle panel, the same macrophages were incubated with a Giemsa stain. Giemsa stain stains macrophages bluish purple, as demonstrated here. To test if these were functional macrophages, we wanted to see if they could eat something. We used a kit that can specifically detect phagocytosis and endocytosis with pH-sensitive fluorogenic dye linked to E. coli-derived bioparticles. The dye discriminates endocytosed from adherent and extracellular particles by turning red in the acidic environment of the phagosome. So we incubated the macrophages with the kit for 2 hours. On the right-hand panel, we see that the macrophages are glowing red, indicating that they phagocytize the particles. Oh, and also in the lower panels, we see marker expression that lends support to the presence of macrophages. We see macrophage markers CD163 and CD68 by flow, with a decrease in CD14 expressing cells. So the iPSC-derived monocytes can be differentiated into phenotypic and functional macrophages. ATCC's iPSC-derived monocytes also have high dendritic cell differentiation potential. To support this, we incubated the monocytes with a commercial dendritic cell differentiation kit and cultured them on attachment culture dishes. On the left, you can see the monocytes prior to differentiation. They have smooth surfaces and are largely in suspension. In the middle and right images are the morphology of dendritic cells on day 7 of treatment. They're showing the characteristics of immature dendritic cells, including a rough surface with multiple pseudopodia, and they've also become loosely adherent. And looking at the results of the flow, like before, we see a decrease in CD14 positive cells along with a concomitant increase in dendritic cell markers CD80 and CD83, indicating that we have indeed dendritic cells. So while it's nice that data shows differentiation, but I'd like to show you an example of how you could actually apply monocytes to something useful. So in recent years, there's been a movement away from using animal-based tests. A good example of this is the various tests for detecting endotoxins, aka pyrogens, in parenteral preparations and biologics. So these compounds can cause a robust immune reaction and fever, followed by severe shock and death. Now, historically, there have been 3 tests that have been used to determine the safety of these parenterals. The rabbit test is the first one I'll talk about. It involves subjecting rabbits to the biologic or parenteral and then monitoring it for fever or death. Bad for the rabbit. Then there's the limulus amoebocyte lysate or LAL method, LAL method, which requires the blood of the horseshoe crab. Either a gel clot is then detected, or more commonly now a change in color or turbidity is measured. Unlike the rabbit pyrogen test, the LAL assay is limited by being unable to detect non-endotoxin pyrogens. And also, since thousands of horseshoe crabs are bled for this per year, and there's probably about a 25% mortality rate of the horseshoe crabs would probably prefer to be left out of this. So finally, there's the monocyte activation test, or MAT assay, which uses the cryopreserved blood of several human donors. While it's much more humane than the other 2 tests, it could suffer from lot-to-lot differences in quality. So you have influencing factors such as lifestyle and genetic background that can differ significantly between the donors, and really we just can't control for that. So we hypothesized at ATCC that since human monocytes from mixed blood are used in the MAT assay, our human iPSC-derived monocytes could be used for conducting the test. And I'll show you that right here. So in the panel on the left, we show a mix of PBMCs. They're ATCC actually, part number PCS 800-011. And on the right, we show ATCC iPSC-derived monocytes. Both are treated with increasing concentrations of LPS. So LPS is a lipopolysaccharide. It's a bacterial cell wall component, and it's highly immunostimulatory. We measured the secretion of interleukin-6, which is a pro-inflammatory cytokine that monocytes secrete when activated by something like LPS. As you can see, the interleukin-6 secretion increases as units of endotoxin increase. Thus, the iPSC-derived monocytes can be used as a humane, inexpensive alternative for conducting the MAT assay. And as I showed you, they have high potential to be differentiated into macrophages and dendritic cells. All right, so let me go ahead and wrap up my presentation now. I've showed you that ATCC developed proprietary methods for the differentiation of CD34 positive cells, monocytes, and MSCs from normal human iPSCs. Something that I didn't mention before, the different ATCC iPSC lines have varied efficiencies for differentiation into distinct lineages. So we commercialized the iPSCs that differentiated the most efficiently. Differentiated iPSC-derived primary cells can provide an unlimited source of cells for your cell-based assays, as well as interexperimental reproducibility and high biological relevance. They exhibit primary cell functionality and genetic stability. I also showed you that iPSC-derived CD34 positive cells can differentiate to all blood lineage cells. I presented data indicating that iPSC-derived monocytes can differentiate to functionally active macrophages and dendritic cells. I also showed that activated cells can be utilized for developing a MAT assay. I showed you how iPSC-derived mesenchymal stem cells can differentiate to osteocytes, adipocytes, and chondrocytes, and they also display immunosuppressive properties. And finally, if you're interested in more information or in ordering these versatile cells, please navigate to www.atcc.org/differentiated iPSCs. Thank you. I'll take your questions now.
Alrighty, in just a few moments, we'll begin the Q&A session. Please use the chat function available through the webinar program to submit your question. Also, Kevin has disengaged, so basically I'm going to be running this solo tonight. So let's go ahead and jump in and start with our first question. And this is actually a pretty good question. Does ATCC warranty include differentiation efficiency of the iPSC? So to answer that, I'm pretty sure, not 100% positive, but I'm quite sure that the warranty covers the number of viable cells per vial, but does not cover differentiation. Now, what I can do is I'll pass this question along to our customer care group and our product line business managers to confirm this for you, and we will definitely get back to you via email. Okay, and for our next question, can you talk about the longevity of the iPSC-derived CD34 positive cells in the context of passaging? And then what about the iPSC-derived monocytes? All right, so it's definitely not recommended to passage these cells. They were designed to be used right out of the vial, hence the high number of viable cells. So they come approximately 3 times 10 to the 6 cells per vial. So you definitely should not try to expand or refreeze these cells. They should be used in experiments immediately after thawing. For the iPSC-derived monocytes, they come at 2.5 times 10 to the 6 cells per vial. And then there's a follow-up question actually that's come into this. Do you have any recommendations for seeding density and thawing media? Okay, so the seeding density is going to be based purely on the customer's application. So it really depends on whatever kit that you're using. If you're doing a particular cell-based assay, or if you're using a particular differentiation kit. However, for thawing the cells, however, we recommend using RPMI 1640 medium with 10% FBS for thawing the cells out. All right, and it looks like the next batch of questions that we have coming in is around culturing the iPSC-derived mesenchymal stem cells. So the first question that we have is, what is the seeding density for the ACS 7010 MSCs? So we'd recommend seeding those cells at 10,000 to 20,000 cells per centimeter squared post-thaw and post-culture. In addition, actually, so the optimal seeding densities are critical for differentiation. So we do have suggested seeding densities in the differentiation media kits for osteocytes and for chondrocyte and adipocyte differentiation kits that we provide. Now for this next question, what is the optimal cell confluence when passaging the mesenchymal stem cell? So, right, the mesenchymal stem cells, the iPSC-derived mesenchymal stem cells are different from the other two flavors of differentiated iPSCs that we have because you can passage them, and really 85 to 90% confluence is optimum. We definitely do not recommend going beyond 95% confluence. And let's see. Okay, this next question actually sort of follows on that. What is the effect of passage number post-thaw on lineage differentiation? So I guess this sort of makes sense. It's generally a good rule with cell culture that you want to use the earlier passage cells if possible, right? So early passage iPSC differentiated mesenchymal stem cells are going to be better at differentiating, than late passage MSCs. So you want to go through passage 1 or passage 2 post-thaw if you can. That's going to be much better than passage 4, passage 5, right? And then another question just generally around ATCC's mesenchymal stem cell offerings. What other mesenchymal stem cells are available from ATCC? So we actually had a table in the beginning of the presentation, and we show that we have adipose-derived and adipose-derived H-term immortalized mesenchymal stem cells. We also have human bone marrow-derived mesenchymal stem cells, which is ATCC part number PCS, I'm going to guess 500-012. And these were the cells that were used actually in the experiment on slide 19, where we showed equivalent immunosuppressive activity with the iPSC-derived mesenchymal stem cells and bone marrow MSCs. Okay, and then interesting. Okay, a question came in around all iPSC-derived primary cells are reprogrammed with Sendai virus. This could possibly affect import regulation, and what is our assurance of total virus zero existence to the regulator? And also, is there a concern for viral components to the customer in the derived cell that makes it non-xeno-free and serum-free environment? Okay, so those are actually very good questions. So one thing that's great about Sendai virus is that it's footprint-free, right? So after the cells have been passaged a couple times, the Sendai virus goes away. And basically, it should affect import regulation to the same degree as the ATCC iPSCs that we've been offering for, I don't know, like 10 years or something like that. So, so I am just guessing right off the bat that if you can order iPSCs from us already, there should be no issue with ordering the differentiated iPSC cells that I showed you tonight. If there are some issues with that, then you may not they may not be available in your area. And then, as far as the viral component in the derived cell that makes it non-xeno-free and serum-free environment, well, originally the iPSCs were passage in serum-free media, serum-free and also feeder-free media. It was not completely chemically defined, however. And after differentiation, depending on which cell you're talking about, there may have been some sera in that. So I can forward this question for a better, more firm answers, actually, to, again, the product line business specialist who's in charge of the products. And they'll be able to give you a much more direct and firm answer on this. But good question. Very good question. Okay. Let's see. Let's move on to the what dissociation reagent is recommended for passaging the mesenchymal stem cells, and is any special medium used? So for this, we used trypsin EDTA for primary cells. So it's a specific formulation that we have. It's ATCC part number PCS 999-003. We also have a trypsin neutralizing solution that pairs well with that, actually, and that's part number PCS 999-004. Okay, and next question. What is the longevity of the iPSC-derived mesenchymal stem cells? Or, in other words, how many population doubling levels and passages can they go? So this is a good question. They can be proliferated at least 20 population doubling levels, or over 10 passages. All right. Okay. Here's a good question that's definitely current and timely. Can the iPSC-derived cells be used in COVID-19 research, and what are the applications? So this is, again, a really timely question. Recently, there's been, it's probably up to 25 or more papers suggesting or reporting the use of mesenchymal stem cells or some kind of stem cell therapy to lessen the effects or improve outcomes for SARS-CoV-2 infection. But right off the bat, our cells are for research use only. That being said, of course, they can be used as standards in the experiments, since we can reliably provide a consistent product. So other things that they could be used for, they could be used to test immunosuppressive capacity. They can be used to study mechanisms for immunosuppression, maybe to evaluate anti-inflammatory responses to viral infection or the cytokine storm. So, also kind of a side note, the angiotensin converting enzyme 2, the ACE2 receptor, it's been identified on hematopoietic progenitor cells. So as we all know by now, that this is one of the molecular mechanisms that SARS-CoV-2 uses to get into the cell. I don't have any data to show you that that's expressed in the iPSC-derived cells, but this would be very interesting to see, and could be another application that you could add this to the list of things you could use these cells for. The CD34 positive cells, they can be used as controls to investigate treatments for the cytokine storm again. There's been some work being done to use CD34 positive cells to help repair lung damage from infection. So these cells could again be used as controls in that purpose rather than a therapeutic purpose, right? For the iPSC-derived monocytes, they can be used in any application requiring PBMCs. So, of course, many inflammatory processes are regulated by dendritic cells and macrophages. So you could differentiate the iPSC-derived monocytes into these cells and then use them as controls to look at immunosuppression. But from the other side of things, you could use them to look at the interplay between monocytes or macrophages and T cells and see how it relates to treatments for SARS-CoV-2. And of course, any of these products could be used to test the safety of vaccines or other therapies. And then kind of as a side note also, we've got a wide variety of primary PBMCs and primary T cells. And, you could also use these to study the efficacy of novel vaccine candidates to activate the cells. And if you were wanting to look at what we had in that, you could check out our offerings at www.atcc.org/cancer-immunology. All right, so moving along to our next question. Are monocytes mostly suspension cells compared to macrophages? And then there's some follow-up to this. Besides the markers, is it possible to differentiate between monocyte stage and macrophage stage morphologically and based on adherence? And then finally, the same asker asks, can we consider activated monocytes to be macrophages when they become adherent and polymorph? So that's a good question, a good series of questions actually. So yeah, monocytes are typically in the suspension where macrophages like to attach to something when they differentiate. So some things that you can test for activation is look for phagocytic activity like I showed in the presentation. Some of the other substrates that you can use would be either like latex beads or apoptotic bodies, which you can get by incubating cells with spirosporine. You could also look at cytokine responses or something like chemotaxis and invasion assays as well for that. All right, let's see. Which mesenchymal stem cell product differentiates most efficiently into chondrogenic lineages? So that's adipose, bone marrow, or umbilical cord. So I actually haven't looked at every single experiment that ATCC R&D did, but what I can tell you is that the iPSC-derived mesenchymal stem cells that came from iPSCs originally were developed from bone marrow CD34 positive cells. And basically, we just selected them for the best overall performance, what performed the best across the board. All right. There's another good question. The differentiation of the iPSCs to CD34 positive cells is not completely homogeneous since at least 20% does not express the CD45 molecule. Why is this? That's it. Someone who's paying attention. It's a great question. So the emergence of CD45 expression actually correlates with commitment to lymphomyeloid cell lineage. Now, once you have a higher proportion of cells expressing this marker, the cells just overall have gone too far along the differentiation lymphoid lineage, and they're pretty much committed then. All right. For our next question, if we generate our own protein constructs that have our own generated mutation, how do we transfect iPSCs or differentiated cells with our constructs? So presumably these cells are transfectable. The parental cell lines are. We've transfected all of them with GFP using our transfex reagents, and we saw the cells glowing green afterwards. So if you're interested in learning more about this, you could go to www.atcc.org/transfection to our transfection tools page. And if you do try this experiment and you have some interesting data to show us, please let us know. Okay, a few more questions that are still coming in. Can iPSCs be differentiated into motor neurons? Okay, I kind of like this question actually. It takes me back a little ways. Yes, they can. If you mean by the TRA160 and NANOG or SOX2 expressing iPSCs. So these are not iPSCs that we've differentiated, not the iPSCs that we're talking about today. But our R&D group, I should say, did actually do that to create neural progenitor cells, which we do sell. We've got a complete system actually of normal and Parkinson's disease trilineage capable neural progenitor cells. So we also have lineage marker labeled neural progenitor cells, and we have an expansion medium. We have a differentiating media that will differentiate the cells into dopaminergic neurons. And we have protocols to differentiate the cells into astrocytes, oligodendrocytes, and to neurons. We also have a ton of application data around this. Gosh, I think it was like in 2018, I presented a webinar on this. So if you're interested in doing this, let me know, and we'll send you a link to the video so you can watch it on demand. The cells are great for doing in vitro neural studies. They really do a good job of getting you to doing your experiments quicker than if you were trying to differentiate them yourself. So, okay, we've got another question here. The various differentiating media from ATCC to differentiate mesenchymal stem cells to bone marrow chondrocytes or adipocytes or monocytes to downstream differentiation. Certainly, we can definitely share that with you. I can send those media in a link to you shortly. Okay, we've got another couple of questions that have come in, but both are kind of specific to our iPSCs, maybe more of a custom service. So the first question is can we request iPSCs to be derived from disease patient donors with certain mutations? And so the answer is definitely yes. We have a very robust donor network. And so to go about doing that, you can contact our custom services via either your account manager or the website under the services tab on our website. And then the second question that's kind of around that is, can you provide differentiated iPSCs created under CGMP or CTCP compliant? And that's actually another customer service, custom service that we could possibly provide. But, you know, I would recommend requesting more information via either your account manager or contacting us through www.atcc.org/services. All right, and I don't see any more questions coming through, so at this time we will conclude our Q&A session. So I'd like to thank everyone for attending this webinar. Thank you again, everyone, and have a great day.